Yeah, let's get started. So we are now in the molecular genetics portion of the course. And so now we're diving into DNA, the A's, T's, C's, and G's, how that stuff really works. So I'm going to start with a brand new Roman numeral. I'm going to start with 1, just because it's kind of like part 2 of the course. The big main heading that we're starting with is DNA. The molecule DNA, otherwise known as deoxyribonucleic acid. So today we're going to go through the history of how DNA was discovered to be the genetic material. We're going to do structural stuff. So get your pens and paper ready, because I love drawing structures, and I want you all to practice drawing it, because I feel like that's the best way to learn the stuff. So first, we're going to hit on some history, and I don't do a lot of history in this course, but it's kind of fundamental. So these are some of the main experiments that were performed historically in the last, let's say, 150 years that have revolutionized what we know about how cells work, how life works, how genetics works. And so we'll start with the history of DNA as genetic material. So we know that Gregor Mendel, he is the father of genetics. He's considered the father of genetics. He was around in the 1860s. I'm never going to ask you a date on an exam, but I want you just to have kind of some concept of the timeline, right? So Mendel was around in mid-late 1800s. And so we've looked at Mendel's experiments, but what he did was he described the laws of genetics and really what those laws were suggesting was that traits were determined by distinct factors. That's the term he used. That physically pass in a reproducible way from parent to offspring. Now, this is in contrast. There was a popular sort of hypothesis around the time that traits were really caused by use. It's Lamarck's hypothesis, and it's something like the example that's often used is like a giraffe has a long neck because it has to stretch up to get the leaves. So Mendel's sort of hypothesis that there are physical units that pass from parent to trait and which are independent of, what you're doing in the environment was kind of a controversial sort of hypothesis. And so Mendel's work went pretty much unrecognized for his life. It wasn't until after he died that his idea of these factors came back to life. Now, he did not know. He did not know what molecules were responsible for the trait. He did not know what the factors were molecularly. He just knew that they were reproducible, something physical that passed on, some molecule that was physically passing from parent to offspring. So he did not know what molecule it was. And he did not coin the term genes either. That was later by scientist Gil Hansen. So Gregor Mendel's work went pretty much unrecognized until the 1920s. And one of the seminal experiments that kind of brought this idea back to life was performed by Frederick Griffith in 1928. So what Griffith did, and so what I'll do for each of these is kind of tell you the conclusion of the work, and then I'll show you just an overview of the experiments that led to those conclusions. So Frederick Griffith gave experimental evidence. You know, Gregor Mendel's stuff was simply statistical sort of analysis. Frederick Griffith gave experimental evidence of Mendel's factors, or for Mendel's factors. So how did he do that? We'll go through his experiment now. So he did what is called a transformation experiment. So in his experiment, he did a transformation, which is changing the traits. He transformed one strain of bacteria into another type. So he did a transformation of one strain of bacteria into another. He changed the traits. He transformed the traits. How did he do it? Well, here are the two different strains. There's an S strain and an R strain of bacteria. So the S strain stands for smooth. It's a smooth strain of E. coli. And it's smooth because it has a lipid coat surrounding it. And it's a pathogenic bacteria that causes pneumonia. And so when this is injected into mice, this smooth strain of bacteria gets into the lungs of the mice, and the immune system can't recognize it because it's got this lipid coat. So it causes pneumonia in the mice, and the mice die. Sorry for the graphic depiction here. So the smooth strain, when injected into mice, causes pneumonia in the mice and ultimately death. The R strain of bacteria, the R stands for rough. Now it is missing that lipid coat. So that means surface proteins on here and different carbohydrates are visible to the immune system of the mice. And so the mice can recognize these bacteria and clear it. And so you don't get pneumonia when injected into mice. So you get no pneumonia because the immune system clears it. And the mice live. Okay, so check. Okay, so the smooth strain, the rough strain. So what Griffiths did was he did different combinations. He took heat-killed S cells. So he took heat-killed S. Now, when you inject heat-killed S into the mice, you've killed the bacteria, and so you don't get pneumonia, and so the mice live. And that makes sense, right? But here's where the sort of magical transformation occurred. If you take those heat-killed S, and live R. Now, neither of these, right? So the rough strain or the heat-killed S do not cause pneumonia. But if you took a living R strain and a heat-killed S, something transferred from S to R to transform those R bacteria into S type. It caused pneumonia in the mice, and the mice died. The mice died, and what were the living R have been transformed into the S. So the conclusion was some factor or molecule transferred from S to the R and transformed it, changed the traits of the R. So this was Mendel's genetic factor. something was changing, was transferring from here to here, some molecule, transforming these into the S-type bacteria. Okay, Griffiths didn't know what the molecule was either. And you know, when scientists were looking at these results and playing around with the ideas, the common thought at the time was that it was probably proteins that were passing from the S to the R, because proteins have a far greater diversity, right? There are 20 different amino acids in proteins. DNA and RNA only have 4 nucleotides. So the thought was, well, it was probably proteins that were changing and transforming the bacteria. And it was actually Griffiths, one of Griffiths' students, Oswald Avery, and colleagues, Colin MacLeod and Macklin McCarty. In 1944, for about 15, 20 years later, they picked up Griffith's transformation experiments and looked at it from a more molecular perspective. What they ended up doing with their experiments, I'm going to give you the conclusion, we'll go through the experiment, is they provided evidence to suggest it wasn't protein, but it was that DNA was the transformative factor. Okay, so how did they do that? What were these experiments? So the experiment here, they expanded Griffith's experiment. So same concept, using the S in the R strain bacteria. So they expanded Griffith's experiments to look more molecular, right? To do molecular analysis of what might be transferring. So what they did was they, rather than using heat-killed S, that was the starting point. So up here, we're starting heat-killed S bacteria. Now these would not cause pneumonia, right? The R strain alone wouldn't cause pneumonia. If you mix those two together, they would cause pneumonia in the mice. But they didn't stop with just the heat killed. They treated the extract. So they busted open these cells and they treated the extracts with different types of enzymes to break down or to eliminate different classes of molecules. So when they mixed the molecules. So the S strain extract, and so this means the molecules, the busting open the cells and releasing the molecules. When they mixed molecules of S with live R, they found that the mice died. It caused pneumonia. So you didn't need to have the physical dead cells. You could just have the molecules from inside S and they would transform the R bacteria. But now if you treated the S, if you treated the S with RNase, ACE is suggesting an enzyme. RNase, if you wanted to guess what this did, oftentimes the name of enzymes tell you what it's working on. What RNases do is they chop up the RNA molecules. So essentially eliminating RNA from the S extract. When they did that, when they took RNase treated S extract and they took it with the live R, they did not get rid of the transformative factor. So the mice still died and still caused pneumonia. You still got transformation of the R strain. So that suggested RNA was not the transformative factor. When they treated the extract with protease, if you had to guess what proteases act on, ACE is telling you it's an enzyme. It's an enzyme that acts on proteins and it chops up proteins. So if they took protease treated S extract with live R and injected it into mice. So now you've eliminated proteins, right? And this was the major hypothesis at the time, that it was proteins doing the transformation. If you got rid of proteins in this S extract, you did not get rid of the transformative factor, and the mice still got pneumonia. So that suggested that proteins were not the factor. But when they treated the extract with DNase, this is an enzyme that chops up DNA. When they chopped up the DNA and essentially got rid of the DNA from the S strain, the mice lived. The mice lived, suggesting that DNA, so it suggested that DNA was the transformative factor. Because when you got rid of DNA, you did not get transformation. So this was the first direct evidence that DNA was the genetic material, not protein, not RNA, that it was DNA that was the genetic material. A follow-up on that, about a little less than a decade later, which really solidified DNA as the genetic material, was done by Alfred Hershey and Martha Chase. Got a question in the chat. Let's see what we got. Was everything already known about DNA at that time? Kat, that's a great question. What was known about DNA at the time was the subunit composition. And what was known about the proteins at the time were the subunit composition. So when we get into the DNA structure at this time, what was known is that you had A's, T's, C's, and G's, but how they were arranged and how DNA works was not known at that time. So the next set of experiments was by Alfred Hershey and Martha Chase. Okay, so great question too, David. So how does DNA go from the dead bacteria to the live bacteria? So bacteria, are not eukaryotic cells. They divide primarily by fission, but it's thought that it is thought that the DNA is actually transferred through these little pili structure, which are kind of like a little channel that can form between bacteria. And so you can get transfer of molecules between bacteria through these little pili structures. It allows for sort of genetic diversity and recombination between bacteria. So Alfred Hershey and Martha Chase in 1952 kind of nailed down DNA as the genetic material. So they gave further evidence for DNA. As the genetic material. So how did they do that? They used not bacteria, but they used a different system. They used a bacteriophage. So the experiment they used here is they labeled bacteriophage or phage. Bacteriophage are viruses. They're viruses that infect bacteria. Here's a little depiction of it. When we talk about coronavirus later, we'll talk some more about viruses in general, but here's a depiction. It looks like this little kind of alien spacecraft, right? With protein coat surrounding DNA on the exterior. So they label these bacteriophage, these viruses, with either or both P32, which is radioactive phosphorus. And when we get into the structure, we'll find out that that's going to label the DNA, so making the DNA radioactive, or S225, which is radioactive sulfur. Now, DNA doesn't have sulfur And proteins, for the most part, don't have as their core structure phosphorus. So the S225 labeled the proteins because the amino acids cysteine and methionine both have a sulfur in them. So they made either the DNA radioactive or the proteins radioactive in the virus. So here it's showing a virus. Here's the protein coat is radioactive, and here the DNA is radioactive. And then they infected the bacterial cells. So this is a bacterial cell here. So what they found was that the proteins did not transfer into the interior of the cell. So they found that the proteins did not get injected, did not enter bacterial cells. But what they did find was that the DNA did. And I guess a key component of this is, I should say, that what they do once they get inside the bacterial cell is they make lots of more copies of themselves, right? So you make lots and lots and lots of copies of the virus, and then it gets shed and goes to infect other cells. So the DNA entered the bacterial cell, the host cell, and was used as a source of replication of new viral particles. So that is the genetic, that was the information to make more viral particles, not the protein coat. I think I've got an animation for this one. I checked this link, it should work.
In the Hershey-Chase experiment, bacterial viruses called phage were used to demonstrate that DNA is the genetic material. The phage used in this experiment consisted of a DNA molecule surrounded by a protein coat. When phage infects bacteria, they attach to the surface of the bacterium and inject the DNA into the cell. The protein coat remains on the outside of the cell. In the first part of the experiment, phage were produced in a medium containing 35S radioactively labeled amino acids. This resulted in a phage population with 35S labeled proteins, but no radioactive label in the DNA. The phage were then allowed to infect the bacteria. The phage attached to the bacterial cell and injected their DNA, but the radioactively labeled protein coat remained on the outside of the cell. The phage produced in these cells contained no radioactivity. The vigorous shaking caused the empty protein coats to be removed, but did not interfere with the production of new phage in the cell. In the second part of the experiment, phage was produced in a medium containing 35P labeled deoxyribonucleotides. This resulted in a phage population with 35P labeled DNA, but no radioactive label in the protein. When the phage infected the bacteria, the 35P labeled DNA entered the cell and could be found in phage subsequently produced in the infected bacteria. This demonstrated that the DNA, but not the protein, carries the generic information for a new generation of phage.
So by the mid-1900s, which seems like a really long time ago, right? But you probably have grandparents or great-grandparents who were around then. So it was about the mid-1950s that, or mid-1900s, that DNA was now accepted to be the genetic factor, and the term genes was being used. So DNA was now the molecule of traits, was the molecule of genetics. And so now we're going to dive into DNA and the molecular structure of DNA. So yeah, back to your question, Kat. At the time, I'm going to tell you, just kind of give a one sentence overview of what the structure of DNA is, and then we'll get into the details. Back in the 1950s, the nucleotides were known, but this structure was not known. So the structure of DNA, it is a two strands, two-stranded chain or polymer of nucleotides. So nucleotides are the subunits of DNA. And those two-stranded polymer of nucleotide subunits, the strands are held together by hydrogen bonds. And it's twisted into a helical structure. So let's dive into the specifics, because fully understanding the structure of the nucleotides is really important to understanding the chemistry of how DNA and genetics works. So the nucleotides. The core structure of the nucleotides, or I'm sorry, the structure of the nucleotides is comprised of three different sort of chemical subunits. All nucleotides contain a nitrogenous base, so here is the nitrogenous base component of this nucleotide. Notice it's called nitrogenous because there's a bunch of nitrogens in it. And it's a base because it can pick up proteins out of solution. So Watson and Crick, right? Watson and Crick were going to do the helix, but these nucleotides were known before Watson and Crick did their experiments. So this stuff was known, that nucleotides contain a nitrogenous base, a pentose, meaning a 5-carbon sugar, either ribose or deoxyribose, depending on whether we're talking about DNA or RNA, and one or more phosphates. So we've got our base component, we've got our pentose sugar component, and we've got our phosphate group. It is this nitrogenous base that is the thing that varies between the different nucleotides, and so it gives the nucleotide, and I'm going to shorthand nucleotides for the rest of the semester, just NT or NTS. It gives the nucleotides its identity because it is the variable portion of the nucleotides. So let's talk about those nitrogenous bases and what they're like. So I don't know, put an A right here. So nitrogenous bases, they give the nucleotides their identity. There are 5 different nitrogenous bases. There's cytosine, otherwise known as C. There is thymine, otherwise known as T. Guanine, G. Who am I missing? Adenine, A. And not in DNA, but in RNA, there is uracil, U. I'm going to say in RNA nucleotides, not in DNA nucleotides. So here they are down here. We can categorize these into 2 different classes. So there are 2 classes of nucleotides. The pyrimidines, and the pyrimidines are C, U, and T. So cytosine, thymine, uracil, and these are characterized by, they have a single ring structure. So there's a single ring, which is what the pyrimidines have in common. The purines are the other class, and they're a more complex structure. There's a double ring structure, and adenine and guanine are the purines. Now, at this point, for this exam, or for exam 2, you're not necessarily going to need to know the different functional group composition, but I'm going to want you to identify to be able to discriminate between a purine and a pyrimidine. You don't need to know the difference between adenine and guanine at this point, or between cytosine, thymine, and uracil. But as we get towards the end of the semester, you'll be able to tell these guys apart too. So at this point, what I want you to know is the difference between purines and pyrimidines, and to be able to recognize which is which. So the base is the thing that determines the identity. It's the thing that differs between the different nucleotides. So that's why when we talk about nucleotides and base pairs in DNA, we talk about C's, T's, G's, A's, and U's. Although there is more to the nucleotide structure than just these guys. So then attached to the nitrogenous base is in DNA a 2' deoxyribose sugar. Is attached to the nitrogenous base. And I'm just going to call it a base too, because usually we don't use the term nitrogenous in there, but that's the technical term for it. So there's a 2' deoxyribose sugar attached to the base. I am going to want you guys to know this structure inside and out, to be able to draw it and everything. So I'm just going to put, let's say this is the base adenine, is attached with a covalent bond to, here is our deoxyribose structure. Now, for those of you who might not have had organic chemistry, I'm just going to kind of talk you through this just a little bit to familiarize. So we've got in this, anytime you see a stick in a molecule, that's a covalent bond. And so here's an oxygen attached to the oxygen. Anywhere that these sticks come together. So there's a question in the chat, does this refer to the nucleotide base or to the nucleotide? This, what I'm drawing here with the base attached to the sugar, is not a nucleotide yet. So we're building the nucleotide up. The base plus the sugar is something called a nucleoside. And so this ribose sugar component is really super important. So that's what I want to draw it out. And actually, let me take a second just for those of you who might not have had organic to draw it out properly. Because there are certain shortcuts you can take when you're drawing out molecular structures. And so this is kind of like a shortcutted. So we have in this ring, we have an oxygen, a carbon, a carbon, a carbon, a carbon, and then we have another carbon outside of the ring. Every carbon has to have 4 covalent bonds attached to it. And so a carbon always has 4 sticks attached. And if it's not showing you what's attached to it, hydrogens are implied. So the base is attached to, in one of those positions, this oxygen, another carbon, and then the other thing that's attached here is a hydrogen. This carbon that's attached to the base is called the 1'. 1'. So the sugar attaches to the base by a 1', and the prime symbol is used to differentiate between the atoms of the base and the atoms of the sugar. So 1' carbon of the sugar is what's attached to the base. And then the numbering from there goes away from the oxygen. Yeah, there's a question, is the nitrogenous base so named because it is basic versus acidic, or because it's the base on which nucleotides build? It is actually has some basic chemical characteristics because there's amino groups on all of those. So it can act as a base. So 1' carbon, 2' carbon, 3' carbon, 4' carbon. The 5' carbon is the carbon that is out of the ring. So there is no left and right in molecule land. There is no left and right in molecule land, so you need to be able to identify these carbons based on where they're at. So the 5th carbon is out of the ring. The 1st carbon is attached to the base. Attached to the 2nd carbon is are 2 hydrogens. Sorry, that 2 is kind of in the way. So it's got to have 4 sticks around it. So there's 2 hydrogens attached to the 2' carbon. The 3' carbon has a hydrogen attached to it in what's called a hydroxyl functional group. So that's a OH. To the 4th carbon, the 4th carbon is attached to the oxygen, a hydrogen, the 5th carbon. And then the 5th carbon has another hydroxyl group. Sorry, that 5' in the way, and 2 hydrogens. So this is kind of the complete chemical structure of the base plus the 2' deoxyribose sugar. It is called 2' deoxyribose because at the 2' carbon, there is not an oxygen. So this is going to be in contrast when we talk about RNA later on. So this is DNA. This is the deoxy component of it. In an RNA nucleotide, I'll just quick draw an RNA nucleotide over here. So this is a DNA nucleotide. It's not a nucleotide yet. This is the deoxy portion of it. In RNA, this sugar is not a deoxy. That's why it's called ribonucleic acid rather than deoxyribonucleic acid. In an RNA nucleotide at the 2' carbon, there is a hydroxyl group. So this is one of the main differences between an RNA nucleotide and a DNA nucleotide. That and the fact that RNA has uracil and DNA has thymine. So you'll need to be able to identify these carbons, whether I draw this upside down, inside out, sideways. Identifying these different carbons is fundamental, the carbons of the sugar, and being able to identify whether it's a deoxyribose or if it's a ribose sugar. So the base plus the sugar is not yet a completed nucleotide. This is called a nucleoside. So we're not quite to the nucleotide yet. If my chicken scratch here, if my artistry is hard to follow, I'm going to switch to a slide that's got the complete structure that I just drew out, showing you all the carbons. So this one is a complete structure. This is the ribose sugar. This is the ribose sugar before the addition of the base. And here's showing you kind of how you would write it in a shorthand way. The base comes in and attaches to this 1' carbon and the hydroxyl goes away. So here's a pyrimidine attached through this nitrogen to the 1' carbon. And here's a purine attached to the 1' carbon. Here's a deoxy, here's a ribose. So the nucleoside is the base plus the ribose or a deoxyribose. The naming of these changes when you attach the sugar. So what was a guanine is now a guanosine. Oops. So guanine becomes guanosine. And it's either guanosine, or if it is attached to a deoxyribose, it's deoxyguanosine. Adenine becomes adenosine. Deoxyadenosine if it's attached to a deoxyribose sugar. Cytosine becomes cytidine. Deoxycytidine. Thymine becomes thymidine. And uracil becomes uridine. And now your uracil would be attached to a ribose sugar, so it would not be deoxy. If a thymine was attached to a ribose sugar, then it would just be thymidine. So the deoxy is telling you what the sugar is doing. This is telling you that you have got a sugar plus a base. So how are we going to convert this nucleoside into a nucleotide? So I said there were three main components: the base, the sugar, and then a phosphate, one or more phosphates. A nucleotide is a nucleoside plus one or more phosphates attached to the 5' carbon of the ribose or deoxyribose sugar. So a phosphate here, I'm just going to draw it out because I think a phosphate is a phosphorus atom with 3 negatively charged oxygens. This here is inorganic phosphate if it's not attached to anything. So this is inorganic phosphate. And we have pools of this functional group floating around in our cells. We have phosphate groups inside of our cells. So this is inorganic phosphate, and we also shorthand that P sub I. Now, when you attach this to some organic molecule, like a ribose sugar, here are 2 negative charges. This R group is just a placeholder for organic molecule. So it's a generic organic. Then it is now an organic phosphate. And we shorthand organic phosphate P with a circle around it. So organic phosphate is P with a circle around it. Inorganic is a P sub I. So the only difference here is that this is free floating. It's not attached to an organic molecule. This is attached to an organic molecule. So let's go ahead and tack it on to the end of our structure. I want to draw it out for you all because I think it's important, like I said. So let's go ahead and let's put a base on here. Let's put guanine attached to the 1' carbon of the ribose sugar. This hydroxyl group that was on the carbon of the sugar is replaced by the phosphate. If we number these, we've got 1', 2', 3', 4', 5'. What we're looking at here is a deoxy guanosine monophosphate. And that's a big mouthful of stuff, right? And so that's a lot to say. So how do we write this in a shorthand? Little d, G, M, P. Deoxyguanosine monophosphate. So we would have DGMP, DCMP, DTMP, DAMP are the different monophosphates. Together, if we just want to say generically, we're talking about a deoxy nucleotide monophosphate, that would be DNMP, where the N is just saying nucleotide, any one of the nucleotides. So how do we name ribose sugars? If we tacked a hydroxyl group on here, this would no longer be, let me put it in a different color. If this had a hydroxyl group, it would no longer be deoxyguanosine, it would be guanosine monophosphate. or GMP. Now I said a nucleotide is one or more phosphate groups attached to the 5' carbon. So how do you think we're going to name, let's name this molecule. This happens to be an adenine base. Do you think you could spell out, give me the complete name for this molecule? And then the shorthand for it. Great, good, I like what I'm seeing. Yep. It's adenine attached to a ribose sugar. It's not a deoxyribose, therefore it is adenosine. There is not one, it's not a monophosphate, it's actually 3 phosphates, right? 3 phosphates. This is adenosine triphosphate, ATP. This is our energy, right? This is biological energy, is a nucleotide. And you know, if you look at all these negative charges, what do you know about charges, charge-charge interaction? Charges, negative charges don't like each other. So these bonds actually hold a lot of potential energy. So when we break these bonds, we can liberate that energy. And this is how ATP acts as our biological energy. These, we can also name these phosphate groups. This is, yeah, they repel, that's right. So we have alpha, beta, gamma phosphates here. And so when we do use ATP as energy, sometimes we take off just this N phosphate and we're left with 2 phosphates. And who do you think that would be? If we had 2 phosphates rather than 3, then we would have ADP. So I think naming nucleotides is super important. And, you know, empowering. I remember, I distinctly remember when I was taking genetics as, I think I was a junior as an undergrad, and I was taking genetics, and when I learned how to name the molecules, I was like, wow. It was so exciting because, you know, we throw around these little symbols and stuff all the time, but structurally it's telling you what the molecule is. So I think it's important to know that. So those are the nucleotide structures, and yeah, these guys were known. So the structure, the atomic composition of the nucleotides, the differences structurally were known about the nucleotides, but how they came together to make the DNA molecule and then how that molecule served as the genetic material was not known. It was not known in 1950. And so there are a couple more experiments I want to tack on here to get to the final helical structure. So around the same time that the Hershey-Chase stuff was going on, and there was all this renewed interest in the genetic material, scientists, a hardcore biochemist named Erwin Chargaff, did a series of experiments, 1949. And here's his experiment. Let's take a look at this. Shows you what Chargaff did was he would isolate tissue from different species, grind it up, do brute force sort of organic extracts, and he would analyze the composition of each of the nucleotides inside those different tissues of different species. So he looked at E. coli, which is a prokaryote. He looked at herring, a fish. He looked at rat bone marrow, and in humans, he looked at thymus, liver, sperm, and he determined the concentration of A's, T's, C's, and G's in all these different species and in different tissues within the species. And what he found was if you looked at the composition of A's and A's and T's and divided it by G's and C's, that the proportion was the same in the different tissue types. And that in humans, A's and T's were at approximately the same concentration, and G's and C's were approximately at the same concentration. Different species had different ratios of A's and T's to G's and C's, but within a species, so here's, let's find one that's quite different than humans. If you look at E. coli, the A's and T's and C's and G's were still similar to each other, even though they were different than humans. So what did that mean? So what, he didn't know what it meant, but what he determined was that no matter the species, no matter the tissue, the concentration of A's was approximately equal to the concentration of T's. And the concentration, these are concentration brackets, by the way, concentration of G was always approximately equal to the concentration of C. So interesting phenomenon, reproducible. What it meant, he didn't know. So in another set of experiments, Rosalind Franklin who was a student in the lab of Morris Wilkins. In 1953, Rosalind Franklin determined the X-ray crystal structure of DNA. So she isolated DNA and kind of locked it into place, and she determined the X-ray crystal structure. Now, I'm not a crystallographer, but basically you have to crystallize the molecule, which means lock it into place, and then you bombard it with X-rays and look at the scatter pattern of the X-rays on a film. And so this means nothing to me, but if you're an X-ray crystallographer, what it would mean is that there was this repeating pattern, and this is indicative of a helical structure. So Rosalind Franklin and Morris Wilkins determined the X-ray crystal structure of DNA. And they determined that it was helical in nature. Now, helical doesn't mean that you have this molecule here. It could be one strand twisted into a helix. It could be two strands. It could be three strands twisted into a helix. So it suggested it was helical, but we're still not to the full structure of the DNA. That was determined, yes, by Watson and Crick. So Watson and Crick used this information from Chargaff and Franklin. James Watson and Francis Crick in 1953 used this information, Chargaff's reproducible ratios and Franklin's helical structure suggestion, to create, they proposed a model of the DNA alpha helix. Now, 1953 to all of us seems like a lifetime, well, it seems like ancient history, right? It really is not. Francis Crick died, I believe, in the 1990s, and James Watson is still alive. So how did they determine it? Well, this is interesting, right? Chargaff's ratios and Franklin's helix. So how did these A's and T's come together to make DNA? So they proposed, so Watson and Crick proposed that the DNA helix was a two-stranded molecule. So two strands of nucleotides held together by base pairing and then twisted into a helix. So let's put the strands together first. So the strands of the nucleotides. These are sticks, right? So if it's a stick, it's suggesting a covalent bond. So these strands of the helix are held together by covalent bonds, and they're very specific covalent bonds. The strands are formed by the 5' phosphate, and I'm going to use shorthand now, of one nucleotide is covalently bound to the 3' hydroxyl of another nucleotide. The bond that forms is called a phosphodiester. Phosphodiester bond. So here's the 3' hydroxyl. I'll call this the 3' hydroxyl of this nucleotide